All right, good evening, everyone. Hello, thank you for coming. I'm Craig White. I'm the acting provost here at UNM. It's my privilege to be here this evening and do some opening remarks and then turn it over to the individual who will introduce our speaker and honoree for this evening. So officially welcome and thank you for joining me this evening for the University of New Mexico's 62nd Annual Research Lecture. Tonight is about celebrating the significant achievements of distinguished Professor Vince Calhoun and to present him with this year's Annual Research Lecture Award, which is the highest honor for research and creativity that the University of New Mexico faculty can bestow on a colleague. The University of New Mexico places a high value on research and prides itself as being designated as a high research activity, or R1 university, by the Carnegie Foundation, a designation specifically for universities actively engaged in research in a broad range of disciplines and fields. But it's not only the case that UNM has a very high level of research activity that produces doctoral degrees in many fields, but it's also true that UNM research is truly excellent, generating knowledge that benefits all of us. Professor Calhoun exemplifies that excellence. Tonight I would like to take the opportunity to personally thank distinguished Professor Calhoun for his contributions and leadership in making research at UNM outstanding. I look forward to his lecture, Discovering Patterns of Promise for Unraveling the Mystery of the Human Brain in Health and Disease. I would now like to welcome to the stage Dr. Juan Bustillo, Department of Psychiatry and Neurosciences and Director of the Schizophrenia Research Program, to introduce our lecturer tonight. Thank you.
Thank you. Thank you. Thank you very much. It's really an honor to be asked to introduce Dr. Calhoun. I'm just going to say a few words. Vince completed his undergraduate degree in electrical engineering at the University of Kansas Lawrence in 1991. He then moved to Johns Hopkins in Baltimore, where he worked and completed 2 masters, one in biomedical engineering and one in information systems. And in 2002, he received his PhD in electrical engineering from the University of Maryland. During the 10 years he was in Baltimore, he worked in the psychiatric neuroimaging lab at Hopkins with Godfrey Perlson, a senior psychiatric researcher. Because of that, Vince, early in his career, had the opportunity to understand the needs of people who suffer from serious mental illness, like bipolar disorder and schizophrenia, and also become very familiar with the gaps, the critical gaps in knowledge that the psychiatric field has faced for many, many years. These illnesses were described over 100 years ago, and they are severe with a high degree of morbidity and mortality. They are common. They affect about 2% of the population worldwide. They're chronic. They're essentially a lifetime disorder. And we don't know their causes or their mechanisms. We certainly don't know how to cure them. However, they're very real. We know they're highly heritable, and they affect the brain in subtle ways and can be managed with some medications that are more or less specific. However, we're far from a cure. The need for a diagnostic test in psychiatry is critical for clinicians and researchers in order to develop new and better treatments. It was in Baltimore in 2001, in 2001, before completing his PhD, that Vince introduced the widely used independent component analysis approach to the brain imaging community. This was only his second first author paper, and it was entitled fMRI Activation in a Visual Perception Task, Networks of Areas Detected Using the General Linear Model and Independent Component Analyses. Vince shared this method in a software package he called GIFT. And since then, this software has been downloaded by over 10,000 users worldwide. When I first became aware of this history, I understood better why Vince in his emails, his name and title is followed by a reference from the author C.S. Lewis that says, the author or researcher shouldn't ask, is it mine, but is it good? Vince then moved to Harvard, Connecticut in 2002 to direct the medical imaging lab at the All-In Research Psychiatric Institute. And he continued to study mental illness. There he got his first R01 in 2005 to develop data fusion methods for fMRI, EEG, and genetics. Data fusion, as you will see in his presentation, is one of the common themes that Vince has pursued. Finally, in 2006, Vince was recruited as Director of Image Analysis and MR Research to the Mind Research Network in Albuquerque. He also joined the Electrical Engineering Department as an Associate Professor at UNM. At that time, MRN was in a serious crisis. It had had 4 directors in 7 years, few grants had been awarded, and only had 3 remaining years of funding through the Department of Energy. Clinical faculty at the Health Science Center, like myself, doubted that the mind would survive and questioned whether we would be able to continue our research in psychiatric and brain disorders at UNM. With the recruitment of Vince and Ken Kill, who joined the Department of Psychology, things really changed. Several faculty became independent investigators, collaborations across colleges and departments in the main and north campus flourished, and a true neuroimaging research community started. Vince is currently Executive Science Officer at the Mind Research Network and Distinguished Professor in the Department of Electrical Engineering. He is the author of more than 500 full journal articles, and 4/5 of this have been completed since he came to New Mexico. He is currently PI of 4 R01s, and he heads a center COBRA grant that he's headed for the last 10 years. And through this grant, a dozen young investigators have received training in neuroimaging and research. And this grant is poised to be renewed for another 5 years. In addition, he leads an NSF EPSCoR grant, which is on the brain imaging and epigenetics of adolescent development. I have been at UNM for 20 years. Every so often, young faculty would approach me and will say, Juan, why don't you use this new scanner or that new MRI at this clinic or at the hospital? And I would always tell them, it's not about the machines, it's not even about the money, it's about the people. Please help me welcoming Dr. Vince Calhoun.
Thanks very much, Juan, for those very nice words. It's a pleasure to be here, and I really appreciate this. It's a great honor to receive this award. What I want to do tonight is talk about the brain a little bit. The brain is the most complex organ in the body, and despite having learned quite a bit about the brain, we still don't know really how it works at the deepest level. One of the ways that we can study the brain is through imaging technology. And so I'll be focusing on some ways that we've been using techniques like MRI to study the brain to try to gain more insights into different mental illness and brain disorders, as well as the healthy brain. So I'll cover a number of different topics, but don't worry, we can finish this well within an hour. I'll start really just with kind of the challenge that Juan mentioned a little bit. Brain-related diseases have, I think we would all agree, a major impact on our lives. And if you look at sum of years of life lost to premature death plus disability, you get essentially you see that mental health-related disorders are near the top of the list. They're even above cancer, cardiovascular disease. As Juan mentioned, there are no clinical tests to identify many brain disorders like schizophrenia. And the real hope is that using some of these advanced neuroimaging tools to kind of peer inside and see what's going on, we can better understand the pathogenesis and try to develop some understanding of the disorders with the hope of helping these individuals. It really impacts us. It's a lifelong impact. 50% of the onset of mental illness happens in adolescence. In this plot here, the green area is mental health, brain-related disorders, and this is through the life, right? So you can see that it's a huge impact relative to all these other factors, including costs. The cost of the care for patients is estimated to be about $6 trillion by 2030. So the Mind Research Network, which is on the UNM North Campus, is dedicated to discover and advance solutions to try to prevent, diagnose, and treat mental illness. So we're doing that fundamental research that's needed by applying state-of-the-art systems, including MRI and MEG, to study mental illness and also the healthy brain. So brain imaging. So just to kind of step back quite a bit, we're not in the old days, prior to, 1895 when you had to cut somebody open, right, to see inside, right? Thankfully, we've gotten past that in cases where you don't need to, you don't have to. This is, I think, around the time of just after UNM was founded, right? X-rays were discovered, and that enabled us sort of a first look. And then in the past 100 years or so, there's been just a rapid advance of medical imaging devices, including modern technology such as magnetic resonance imaging. And through this kind of a scanner, we can basically use different techniques to measure these beautiful, basically the wiring of the human brain, without, completely non-invasively. We can see which parts of the brain are activating or are engaged, or are depending on whether you're in doing a specific task, or even if you're just resting there, how they're functioning. And we can also use that kind of information to look at how the brain's connected, how the different regions interact and connect, and we get networks out of this, which gives us a lot of information. I wanted to give just a couple slides. I'm going to focus mostly on functional magnetic resonance imaging tonight. And for those of you that aren't familiar, there's really three kind of fortuitous things that enables us to see inside the brain with fMRI. The first is we have oxygenated blood coming from the heart into the brain, and here's where the neurons are in the capillaries. And then you get deoxygenated blood going out. And when neurons fire, you get this localized increase in blood flow, right? So the blood flow is different here than it is here, right? So that's the first thing. Secondly, your blood happens to be magnetic. So hemoglobin, heme is iron, and so your blood has basically a magnetic property to it. And thirdly, when oxygen is attached to your blood molecule, it's essentially non-magnetic. And then when oxygen is detached, when it's delivered, it becomes magnetic. So it changes its magnetic signature based on whether it's carrying oxygen or not. And so that is what lets us see, all of those things together let us see brain activity with MRI. Basically, we have oxygenated blood coming in, and then the oxygen gets used, and then we produce these magnetic blood cells, which produce a magnetic field, right? So we have a magnetic field. And then when neurons fire, you get this increase in blood flow to that site, and so you have less oxygenated blood there, and you have a lower magnetic field. And then if you subtract these two, you see a difference in the regions that, where we have brain activity. So all of that gives us a really powerful technique, and it doesn't require any injection. It's completely non-invasive. It's completely safe. Another piece to the puzzle here is smarter computing, right? So big data is a buzzword you may have heard. There's a lot of big companies that are analyzing data through different algorithms, bringing them to bear. And if we think about this in the context of the brain, well, we obviously want to use brain imaging to see what's going on in the brain, right? That's a natural thing. And if we think about big data, well, more data gives us better results. We can see more of what's going on in these patterns. So let's collect a lot of brain data, really fine resolution, lots of time. But the problem is that we end up being this person underneath a pile of data, right? So we've basically overwhelmed our systems, and we need to, it's hard to figure out what the healthy brain is doing, let alone when you get some complex brain disorder or mental illness that changes the brain in very subtle ways, and it's very hard to understand all of that. So what we really need are to marry this very sophisticated data with methods and algorithms that can help us extract meaning from the data, simplify and understand what's going on. I always like to show this slide to kind of illustrate that. If this is elastic scattering data, and suppose I come to the data with a very simple model. I'm in the shower one day and I have a eureka moment, and I think a great model for this data would be the backs of elephants. Right? So I fit that to the backs of elephants. I'm really excited. It fits. I even go as far as to predict my next data set. I have a science paper now. I'm excited. It's a silly example, but it kind of gets drives home the point. Remember how little we know about the brain, and the models. Not that we shouldn't bring models to bear. We absolutely should. But the models that we're using are so simple and so imperfect. That we can really get misled unless we let the data tell us more about what's going on in the brain. And a few quotes to drive that home. George Bach said, "All models are wrong, but some are useful." If we had a perfect model, we would have the thing itself, right? Models meant to be kind of a simplifying set of rules that we learn about something. And my favorite is, I believe in ignorance-based methods because humans have a lot of ignorance, and we should play to our strong suit. So. Which is what I'm doing, I guess. So, and you're giving me an award for it, right? So no. The other thing is, really a focus on patterns. So I really think that's important, looking at the whole brain together. How many people see something in here besides rocks? Any of you? Any of you? Raise your hand. No, a couple, right? How about if I do this and back off, right? See, there's a person in there. So the idea is that there really are, we hope, patterns in these brain imaging data that we can use algorithms to find, and they're not just, magical reorganizations of data. They're actually telling us something structured about the data. So a lot of what I do is also looking jointly, not just at individual points in the brain, but looking at them jointly together, right? Or looking not just at one data set, but looking at multiple data sets together. And a simple motivation for that is, suppose we had a brain volume data set and a brain function data set, and there are two groups. Maybe there's a patient group and a control group, and neither one of those data sets can see, the difference between those groups. But if we combine that data together and look at it in this direction, we start to see some differences between the groups, right? So that's the idea. All of these imaging modalities are imperfect, incomplete. They're just snapshots and telling us some information about the brain at a very high level, right? And so we want to leverage all of that data as much as we can. So I tend to use also a lot of basically they're called multivariate approaches. So instead of looking at individual points one by one, we want to look at the full pattern, right? So we can pull out things like this little pattern here that exists within that data that you would not see if you looked at them individually one by one, most likely. So finding the individual is really about classification, right? So it's a much more stringent criteria than just saying I've got two groups of folks and I just want to see if there's a significant difference between those two. That would be looking at differences in the distribution, but I want to say something about an individual person, right? Not about a group necessarily. I mean, they're both useful, but they're different. As Yogi Berra said, prediction is difficult, especially about the future. And this quote is attributed, I think, to several other people, Mark Twain and somebody else. I can't figure out who the original source is. So just to kind of drive that home, if you focus on the right of this slide here, there we have two different points, clouds of points, and they're actually, the mean of those, the average of those is significantly different. If you do a test on it, you get a significant difference, right? But classification, if I want to say something about an individual, it's not very high. It's about 60%, right? So they're overlapping quite a lot, right? Here we have a really identical mean between the two. It's not significant, but these can be easily classified. Right? If something's high or something's low, I can pick group 2. And here's a case where they're the same. Both of them are different, both the mean and the individual. So just driving home the point, the bulk of studies to date have been focused on groups. Let's look at groups of patients. There's been less of a focus on the individual, trying to turn this information around and say, what can we say about an individual person? There have been a number of studies, though, in the field. This is a recent review we did. And here you can see kind of some of the accuracies. This is sort of 60 to 100%. ADHD, major depression, autism, schizophrenia, Alzheimer's, and mild cognitive impairment. And with some success. We're kind of getting in the 70s and 80s. Some of the studies are a little bit higher, right? We still need to do better in some cases. But a lot of these studies are also not done in a way that can really generalize to a new data set. They're kind of telling you something about that data, but they're not able to go beyond that data set. And that's one of the reasons that there needs to be more focus on these kinds of individual subject predictions. And this slide is just to say that we need to do more work on developing classification algorithms. This is one that's widely used, and there are a ton other ones. There's some better ones as well. But support vector machine, all you're doing is you have basically a cloud of points, and you're trying to train it to say, well, let's draw a line through these points, and it can be a nonlinear line, such that we maximize the distance between the closest point and that line, right? And you're trying to find a rule that can separate these subjects from one another so that you can then apply it to a new data set. And another theme in my work is again, it's trying to maximize the information that we're getting out of this. It's to go beyond just looking at the mean or average. So we know what the average is. It's just the sum of a bunch of things divided by how many things we have. And the variance tells us how spread out the points are, right? So those are statistics, right? But those aren't the only statistics. You can keep going, right? This is a first order statistic. This is a second order statistic. And you can go up to skewness. Am I tilted to the left or the right? It's Republican and Democrat, maybe? I don't know. Kurtosis, is it pointed or flat? And so the techniques that I use, and Juan mentioned independent component analysis, is really trying to leverage the higher order statistical information in the data to get at these patterns and to extract them. So let's just think about that a little bit. So we've got a lot of things I've been talking about, and they sound fairly complex, right? And what we're really interested in is a classifier, something that can discriminate, between subpopulations and characterize an individual, and also that we want it to be able to be replicated. We don't necessarily need the data that we're looking at to be robust by itself. We basically want, we're mainly interested in that individual prediction and replication. And higher order statistics, of course, it's harder to compute, right? They're more complicated to compute. But we shouldn't avoid them just because we think that's going to be harder, right? And just to kind of illustrate here from a paper by Eric Earhart, we have the mean map. This is predicting age. If I want to just predict your age, right? I could ask you. That's probably the best way to do it. Well, maybe not for some of you, but I can scan your brain, and I can get near 100% just by taking the average of the volume of your brain, after accounting for your head size, right? I can get very well because your gray matter kind of reduces a little bit as you get older. It's very good. That's all I need. I don't need any more than that, right? But if you look at something like schizophrenia, well, the mean map is giving us, about 50%. But I can get significantly higher than that if I use a more complicated approach. And so the idea is, depending on the question you're asking, the approach may need to be more complicated. You've probably heard of Occam's razor, right? So if you have two equal solutions, the simplest one is the one you should pick. But that is often used as an excuse to say complex things are bad, right? And that's not what Occam was trying to say. And it needs to be tempered by Carl Menger's law against miserliness. He's got a paper that actually talks about that. He's a mathematician. And basically, entities must not be reduced to the point of inadequacy. Okay? So simple by itself, inadequate, not good. Okay. Ultimately, replication is the gold standard. We want things to replicate. So let's take all this and move forward and look at thinking about patterns in our data. So this is an example of independent component analysis, what you can do with it. It's a Halloween example. I didn't have anything for the summer, so we're using Halloween. We have a video of 3 pumpkins on a step. And what we're trying to do is find spatial patterns that are maximally, independent of one another. And so if we take ICA and analyze this, we get out patterns that look like this. Here, one is kind of the background image, and this is the time information. This is how it's changing over time, this pattern. Here we get 3 candles in separate components. And you can see really quickly that candle 3 went out about halfway through the night. So it gives us a really nice high-level summary of a complex video. Right? fMRI is kind of like a video. We're taking images over time in the brain. And so we use this a lot with fMRI data to produce things like this. Okay? So what we get here is, this is people in an MRI scanner. We're collecting a bunch of MRI data while they're just kind of sitting in there, right? And we're looking at how their brains are connected. The different parts of their brains are connected. And what we can get out with ICA is these different networks. Each set of 3 here is a different network that we're pulling out in the brain. And what that tells us is the regions here that are bright are sort of communicating with one another. They're correlated together. So these gives us a bunch of different networks. And if we organize these by their anatomy a little bit, here we have sensory motor related networks. These are in part of the brain that's called the visual cortex. This is a network called the default mode network. It's labeled that because it tends to be more active when you're not focusing on a task or something external. It's kind of your zoning out network, perhaps. And so we get all this information, but we also can look at not only these individual networks, but we can look at how they communicate with one another by looking at this over here. And just to kind of explain this a bit, each of these networks is represented as one of the rows of this matrix. And what you can see here is, for example, these visual networks have a bright red block here. So they're correlated with one another, right? So the visual networks are talking to one another more than they are with the other networks, which kind of makes sense. Over here is your default mode network, and it's also, interestingly, negatively, or it's anti-talking, right, with these other networks. And that's also something that's well known. Okay, so that's just to give you a picture of all the information, some of the information that we can get out of these data. So let's look at the networks themselves, the spatial patterns, and how we can apply that to study mental illness. And so one of the reasons I like using this, again, they're multivariate, and so they tend to be more robust than techniques that are just looking at individual points one at a time. So here we have a component that has sort of bilateral temporal lobe regions, and this is a default mode component. So these are 2 different components. And this is a univariate analysis, basically looking at individual voxels and getting a map out. And we're looking at just a small, just about 50 seconds worth of data here. Or we're looking at, we add more data as we go to the right, right? And what you'll notice is, look at how stable these patterns are, even with just a little bit of data, right? So they're pretty robust. Whereas this one here, it's kind of all over the place. It's going out, then it's coming in, it's going out, and it's just very sensitive because it's only looking at a portion of the data at each point in time. So this basically led us to say, well, let's see if we can use these as predictors, right? Can we take these maps and estimate these from individuals and say something about what's going on? So this is a study we did looking at schizophrenia, bipolar disorder, and healthy individuals. And so the way this worked, this is a pretty simple approach. We basically, because we wanted to see how well it worked on subjects that the algorithm didn't see, so we took out 3 subjects, and then we took the remaining subjects, and we basically computed the differences in their maps, right? So this is healthy control minus schizophrenia, healthy control minus bipolar, schizophrenia minus bipolar for temporal lobe and for default mode, okay? And so once we get these, we freeze those regions and we say, well, those regions are where things are changing, right? So let's take those regions and go back to the original data. We can develop a simple rule that says, if it's greater in these regions, it's likely healthy, healthies are greater, so it's probably a healthy control, et cetera. And when we do that, we actually get around 90% accuracy in predicting, these groups, the schizophrenia patients, the healthy controls, and the bipolar. Okay, so that was an exciting result that sort of encouraged us to keep going on this trajectory. This is a neurological result. This is looking at mild cognitive impairment. So just I'm going to show you some different examples of how we use this. And so this is the default mode network. And in that network, you'll notice that people that were that eventually converted to mild cognitive impairment, but they were scanned prior to that point, versus those that never converted. And you can see that you get a much brighter network here than you do in those that convert. So this is sort of a predictor, an indicator that they might eventually convert to being cognitively impaired. And here you can see, if you compare Alzheimer's disease to converters, those are kind of on the lower side here, whereas up here on the higher side we have the controls and the non-converters. Okay? And getting a little bit at the data fusion angle here, we have additional information. And if we throw more information in, we can often get better results. So here, this is a particular kind of plot, but basically you get better results. You want to have the curve be sort of as far up in this corner as possible. That gives you the best prediction. And if you notice this light green curve, it's the curve that includes MRI plus a PET scan plus a CSF measure plus a number of different covariates included, different variables that we've included, gives us about a 92% predictor. Whereas we get, decent but worse prediction if we use less of the data. Okay? So that's kind of the main message. Here's an example from some work by Yu-Hui Du in my group, looking at psychosis. And so one of the challenges with, as Juan mentioned, that the diagnostic criteria are about 100 years old, and we don't have a lot better criteria these days. What we do know is, the categories that we do have are schizophrenia, bipolar, healthy control, but there are also these folks that are schizoaffective, which maybe they've got some symptoms that are close to schizophrenia, but they've also got some mood, maybe depression, right? Or possibly mania, maybe they're manic. And so these are the best categories we have, and everyone knows that they're imperfect. But using brain imaging data, we take these networks and we can start to see, this is a hierarchical clustering based on those patterns. And you can see some interesting features. Indeed, healthy controls are closer to bipolars, but they're in different trees here. Whereas over here, we've got all the 3 schizophrenia-related categories, and the schizoaffectives are closer to one another. So there's some indication that these categories are giving us information that's sort of reinforced by this biological data, but it's not perfect. Indeed, there are a number of subjects that sort of fall on the boundary, that are in different categories. And so a lot more work, and there is some work now being done, needs to be looking at those boundary cases and really talking to the patients and really trying to understand, what is, why might they be there? Why is their brain information telling them they're on the boundary there? And how can we use this information to refine these diagnostic categories? There's lots of discussions about this. This is yet another topic. This is some data collected by Andy Mayer looking at traumatic brain injury. So again, it's a very different thing, but we can also use these patterns to predict brain injury. Indeed, we can get about 80% accuracy if we look at the fMRI data. And that's not the only information we get. So we always need to go back to these patterns and see what they tell us. So for example, we see that the connectivity the predictive connectivity is mostly occurring in the relationship between cerebellar regions and these long distance connections to the cortex, right? So here we have a post-central SMA region, which is impacted, during that damage. So again, just another example. And So that's been some information about the spatial patterns. We haven't talked much about the temporal or the timing information. We can use that as well. This is a study that it was run by a colleague of mine, Tom Eikola. And it was people in the scanner, they're doing this really boring task, right? It's just you're pressing a button, you know, when things match or don't match. After a while, you just get so bored, you kind of zone out, and then you make an error, and then you focus back in on it. And what he found was that these regions that are active during the task, here's an error. Here's when an error is committed. They start to get less active up to 30 seconds before they commit that error. That's kind of the zoning out, right? And paired with that is that default mode network. I told you that one was more active when they're kind of not doing anything. That one becomes more default prior, you know, to engaging the task. So as often happens, you know, this gets into the press, right? And so they embellish a little bit. So one of the articles called this the brain fart study. And so, you know, forever since that point, my poor colleague Tom is, you know, I know him as the author of the brain fart study. So, and so, you know, up here, this is an example from a fairly large study of schizophrenia. And up here we have all the different networks. We talked about the spatial patterns before. But if we focus on the temporal information, you know that matrix I showed you before, we can see here the healthy control and the schizophrenia matrix. And you can see visually that there are some differences there. This is the subtraction of the two. And there generally is, in a lot of different, you know, regions, there's reduced connectivity between these networks in the schizophrenia patients. It's always nice to try to make sure that you're not, you know, doing something in a way that's kind of biased or that reinforces your own beliefs. And so one of the ways we tried to do this we took a bunch of our data and we threw it online into this competition website called Kaggle. And we basically designed a schizophrenia classification challenge. We basically said, okay, all you machine learning geeks, come to this data and see if you can figure out who's who, you know, in the data set. Me being one of those geeks, right? And so it was actually, you know, quite a successful study. We actually got over 2,200 teams submitted entries to this. Every dot on here is an entry. And the way Kaggle works is you have this leaderboard, and you basically get some data, and you can train your algorithm, and then because you know who's a patient and who's a control, and then you get another data set, and you don't know. We don't tell you, right? And you just have to submit your guesses, you know, based on your algorithm. And so then it tells you after that how you're doing and who you're, you know, which team you're beating, who's at the top of the leaderboard. And so this was really quite fun because we saw on the chat board all these teams were kind of talking to one another, and they were talking about conspiracy theories, and are we cheating them with, you know, trick data in here, and you know, et cetera. You know, and the final winner is determined by a private leaderboard using data that they've never seen at all. And so the encouraging thing was that the cloud of these points is centered well above 80%, right? So there is clearly information in here that others are finding, even close to 90% for some of them. But that's not the only lesson we learned here. We also learned, we probably already knew this, but every individual algorithm is flawed and missing something, right? So if we basically take sort of a pool of experts, we basically take, say, the top 10 algorithms and do a voting scheme. So that, you know, if 6 out of the 10 vote that it's a schizophrenia patient, then that's what we label it. And we find that gives us better performance than the winner in all the way up to the top 65 submissions, right? So we still have a lot to learn in developing techniques, no matter, and all of these algorithms were optimized, and they probably did use lots of complex things, right? Okay, so a little deeper into the timing information. One of the things I like about working with brain imaging data is that we're always discovering new things, and there's always another layer that we missed before, right? It's pretty interesting. But this is one that's kind of come on the scene in the past 5 or so years. I talked about sort of connectivity, but all of the connectivity work was really focused on something like this, where we're saying, okay, these guys are correlated, at 0.4, right? But clearly in this sort of toy example, we're missing something. We're missing the fact that it starts out highly correlated, 0.8, and then they're non-correlated, sort of 0.1 at the end, right? So there's this interactive information, which actually is probably in our data, right? The brain is very dynamic. You're in the scanner, you're doing lots of things, probably that the investigator didn't want you to do, thinking about why you're in here, or I've got to go to the bathroom, or I forgot to do something on my project, et cetera. And so we started to look at the data in a way that captures dynamics. And so instead of computing a connectivity matrix here using the whole data, we just use a portion of the data. And then we compute another one on a different portion of data. We do that again and again and again. We get this sort of time-changing connectivity, right? And so once we do that, you can see some examples here of what that looks like over time in a few folks. But once we do that, now we have this sort of time series of connectivity matrices. We could do something pretty simple like cluster them down here. So we basically, in this case, have what we think are sort of 5 states, right? There are 5 connectivity patterns that they're not in all the time. They're going into this pattern, when they're doing something, and then they go into another one, right? So it's kind of dynamic. It changes over time. This is an example from one subject who started in state 3, then state 2, then state 4, et cetera. Okay? This is the I've given up state, kind of at the end. I'm really tired. And so using this technique has really revealed a lot of information. But when we first start doing something new, we wonder, are we just making up a story? Is this a pattern that we're just, the brain is very good at finding patterns in data, and sometimes they are meaningless, right? So we try to find ways to validate. And so one way we did this was we have a sleep stage study here. We've got EEG data collected simultaneously with fMRI, and these people are falling asleep in the scanner. And we can use the EEG measures to do sleep staging on them. And then we can link that to these dynamic connectivity patterns. And what we find is that one state here is state 1 is strongly associated almost uniquely with the most awake condition, whereas state 5 is associated with the deepest sleep state. And in these, we usually see decreases of these blue regions in there. So that's one way. This is one subject, and this is their hypnogram in green, and this is their state vector from the fMRI in blue. And you can see it matches fairly well. Another way we can get at this is replication. So we have access to a large amount of data. This is data from 7,500 subjects, and or 7,000 subjects, and we split them into 28 independent groups of 250 folks. We analyzed each one completely separately, okay? And then we asked, if we compute dynamic states from these, do we see any similarity between them? These are all different individuals. Is there some canonical state information that we can find? And the answer is, indeed, there is. I mean, we find the patterns match quite well, correlated around 0.8. And not only that, if they spend more time in one of the states in one of the data sets, the match state in another data set, they also tended to spend more time in there. So there were a lot of matches like this. This just gives you kind of a quick visual depiction of that. Basically, these are 7 out of the 28 data sets. And these are the states that we found, and you can see kind of how similar they look across these very independent data sets, with the exception of state 5, which kind of flips back between two different states. And you can see how they cluster together. It's fairly noisy, but we also get very clear sort of centroids when we do this, so that they've got sort of clear portions of non-overlap in these clusters. Okay, so let's go back to our schizophrenia data set that I showed you earlier. We had already analyzed, but we can go a layer deeper, right? So here we're looking at states. We have 5 states from healthy controls and from patients, and this is their difference. And the first thing you note is that we get things that show up in certain states and not others. All right, so these are basically subcortical and auditory and then sensory motor regions that have been implicated in schizophrenia before. hitting state 2 and state 3. Okay? But even more interesting, we can basically calculate how much time individuals spend in each of these states. So here, schizophrenia patients in red spend significantly more time in these relatively weakly connected states 4 and state 5, whereas the healthy controls are spending their time here in states 1 and 2. So we've gone from patients are just less connected, they're showing less connectivity, right? To no, that's not quite true. We're actually, they're actually showing periods of high connectivity, but they're showing they're spending more time, periods of time, in weaker connected states, right? So it gives us yet another layer of information that could be really useful. And this dynamic information is also more predictive than the static information. This is looking at, again, schizophrenia, bipolar, and healthy control. But if we just take this sort of static connectivity and we develop a classifier, we can get about 60% accuracy. So it's not bad for 3 groups. 33% would be your chance level. But if we use this dynamic information for multiple states, we can get upwards of 80 to 90% accuracy. So again, it's much more predictive because it's probably a more natural way to analyze brain data, which is very dynamic. And this is a recent test we did, some work with Jing Sui, where we're working with a colleague in London, Ontario, and she has a clinic, and she works with patients who, she works with major depression and bipolar disorder. And she was telling us that she had a number of patients that early on in the course of their treatment, she was having trouble telling whether they were bipolar or depressed. And only basically months to years later was she able to figure out what the right diagnosis was. And so imagine the amount of time that these folks are on the wrong medications, right, in terms of trying to treat them. So what we did was we took some data from bipolar and major depressed, and we trained an algorithm and found these patterns in the data that were predictive of those groups. And then what she did was she sent us a dozen subjects, and she didn't tell us what the diagnosis was, right? And so it's always good to kind of have these checks built in. And so we took those patterns and kind of like on the Kaggle site, we developed, we decided what the prediction was, which group they're in, et cetera, and sent those back to her. And then she was, her email back was stunning results. She was really excited because we basically got all of these correct, with the exception of one individual who she thinks actually has schizophrenia or schizoaffective. And so, now we have a map of specific networks that we think are predictive of these disorders. But I think we need a lot more test cases like this, really trial runs, kind of see how we're doing. And we also look at the normal brain, right? So that can tell us a lot as well. This is work from Ashkan Fagheri, who's a PhD student in my group. And we're looking at a data set of ages 3 to 21, okay? And so this is age here, and we have 2 states, one that's kind of more connected and one that's more weakly connected. And you see that over time, people are sort of, as the older people are spending less time in the weakly connected state, and they're spending more time, we're getting an increase here in this highly connected state, right? And this is the difference between them. Similarly, this is some data from the Generation R study. It's a Netherlands study. They're scanning all their kids, longitudinal over time. But this is a data set of about 800 kids between ages 6 and 10. And work done by Barnali Rashid, who was a graduate student, and now she's a postdoc in the group. What we found was that the younger kids, again, like that previous data set I showed you, spend more time in this less modular state. And as they get older, they spend more time in this state. But then we went and looked. In this data set, they have a measure of autistic tendency. It's called the Social Responsiveness Scale. And we started to look at that, and we found that the kids with more autistic traits were spending more time over here, right? Regardless of age. Whereas the ones with fewer of those autistic traits were spending more time here. So again, pretty interesting result, giving us some clues about where in the brain we might want to be looking. Okay? And, just to briefly mention, it's really complex. There's a lot of different factors that we have to consider. Substance use is one of them. Schizophrenia patients tend to smoke a lot, right? So we don't want to just be predicting they smoke a lot, right? That's not useful. So we marry these studies with other studies where we're looking at substance users and trying to tease apart the impact of alcohol or smoking or marijuana, et cetera, and finding which patterns are predictive of those in order to try to see if we can control for those in the schizophrenia data sets. Okay, we're almost there. We're getting there. Hang in with me. So I know you guys already ate, so you're probably pretty sleepy. But so deep learning is another buzzword that has been used a lot. For those of you that aren't aware, it's a technique which is, it's what underlies Siri and the Amazon device that can order your pizza, things like that. And the self-driving cars, stuff, all of those models. Essentially, they're more complex models. You basically take a model and you stack it, and you can keep doing that. And they do it sometimes hundreds of times. It lets you model more subtle and complex things in your data. This is an example, and so our question was, is this going to be useful to study the brain, right? We weren't sure, so we took a look at it. And what I'm showing you here from left to right is essentially no model. We're just taking raw data versus this is kind of where we've been. This is working with a single layer model, 2 layers and 3 layers. And you'll notice kind of pretty strikingly, the points are starting to pull apart, and we have a patient group and a control group in those models, and they're starting to pull apart from one another, suggesting that we're increasing our predictive power by using these techniques. Another interesting result here from a Huntington study that we did is, we have controls here and we have patients here, and we didn't tell the algorithm anything at all about disease severity. But you can, it's a genetic disorder, and the more CAG repeats you have, the more severe your disorder is. And so after we did the model fit here, this is a deep learning result, we color-coded. Each dot here is a subject, right? We color-coded them by disease severity. And you see this gradient here where we have least severe by the controls up to most severe, right? So that's really powerful because the algorithm just found that in the data. We did not know what to look for or tell it to find that. So it's not biased at all. It just kind of discovered it. I don't know how often this will happen, but it suggests that these techniques can be really powerful when we bring them to bear on brain imaging data. And we can use these kinds of visualizations to help us. The sort of dirty little secret that we have is that despite buying the same scanners, you do scan people on different scanners, there's probably some scanner-related differences in there, right? And so we can study that if we can amass large data sets and start to see if we can look at calibration, for example. So this is showing you 10,000 data sets collected from 2 different MRI scanners, one that's 3 Tesla and one that's 1.5 Tesla, different field strengths. And, okay, they don't overlap, right? You kind of would want them to overlap with one another, right? But if we look over here, we color-coded them by the age of the subject. And notice that we get these very similar trajectories going from the youngest, 0 to 9, up to 70 plus, right? And then on the 3T data, the same. Those are pretty similar to one another. So suggesting that, in this case, we could almost shift over and calibrate those scanners. We need more, more information like this to be able to do that. But it really gives us some useful information. So then talking about, that gets at the point that we need lots of data, right? So thankfully, the field has recognized this, the imaging community, and there's been a lot of effort to share large data sets. We have a tool called Coins that we developed, which is used around the world. It's used here and in many other places. And it does multiple things. It's a study management tool that can kind of capture all your data, your imaging data, your questionnaires, et cetera. And that's all done within the context of encryption and individual study protections. But once you do that, you no longer have the case. When I first started doing brain imaging, one of the things I hated was that people would do a scan, they'd take a CD, and they'd go away. And then maybe you'd write a paper about it, the CD would go in the drawer, the data would disappear forever, right? This lets us put all the data in an integrated system. It's all protected, and it's all got its permissions based on individual IRBs, et cetera. But if someone wants to get permission to share data with another study or with the outside world, they can do it by pressing a button, right? Or by integrating with people. And so here you can see some examples of that. We have something called a data exchange, which is sharing some open data that we have, a subset of this data. And this is the data that has gone into coins in red, and blue is the data that has been shared, so around the world. So people have downloaded or uploaded data from around the world to this system. That's a centralized model. Another model is a decentralized model. So we have another tool where maybe somebody has a data set and they can't share it, right? It's a rare disease, maybe there's identifiability issues, or their IRB won't let them, or they don't want to, right? I just don't want to because I want to write stuff about it for myself. But I want to help contribute to something you're doing, so we have a technique that lets people you download this tool and you can map to your data, and then basically someone can create a consortium by recognizing all that data. The data never leaves the local site. What it does is you create a model, and the model goes to the site, and it runs a model on that and computes some summary measures, maybe adds some noise to it, and then sends that back, not the raw data. And using this technique, I think, will be a nice complement, because there are a lot of cases. You have international boundaries, you have multiple large studies that have various restrictions that have to deal with it. Just one example of how it's powerful. Here we have sort of 11 different data sites, and they're asking a question about classifying two groups, Group A versus Group B, and they're getting an error rate pretty much across the board of around 25%, because they don't have a lot of data at each site. They can't train their models very well. But if you use this decentralized approach, again, protected through privacy. We're using techniques called differential privacy, which is used to protect health records. It's it's a probabilistic protection of your of the data. So you can't reverse engineer. You can't even run a machine learning algorithm and predict from that data who that came from. And it drops the error rate here to about 5%, which is very close to what you get if you had all the data locally. So this is, I think, a really powerful technique. Okay, last topic, getting there. Juan talked about data fusion. This is my son Jacob doing data fusion. He's 14 now, and I'm glad he's not here because he would complain to me a lot about this picture, but I still use it. But the idea is, hopefully, we're doing it a little better than he is here. But the idea is we're trying to marry complementary information. So EEG data, for example, gives us millisecond level accuracy on timing, whereas fMRI gives us spatially localized information about blood flow. They're different, right? But they're complementary to one another. So an example of this is, this is a story where we have a guy, he's kind of drunk. He's under the street light there. He's kind of kneeling down and moving around on the ground. Police officer comes up and says, what are you doing? He says, the man says, I'm looking for my keys. And so the police officer then asks him, well, where did you last see your keys? And he points way over there into the dark, right? The police officer's like, well, why are you searching over here? Well, because I can see better over here, right? Obviously. This is a brain imaging technique, right? We can see certain things, but maybe we can't see other things. And that really motivates data fusion, right? They're all incomplete. We can get information about white matter, the wiring of the brain, the structure, the morphometry, the shape of the brain, its volume, brain function, what's active, electrophysiology, EEG and MEG, and genetics as well, or behavioral data, or cognitive data, right? And really, we should be bringing all this together. We should, but it's complex. This is a really early study we did that drove that point home for me. These people are doing two different tasks in the scanner, and they'd typically be analyzed separately. If we just focus on the left here, what we do is we have an algorithm that creates these distributions where we're seeing task 1 versus task 2. And each dot here is an individual subject. And these are patients and these are controls. And you can kind of see some differences there, right? You see the controls tend to be sort of flat like this, right? Whereas the patients are giving much more of an angled, right? So what does that mean? That means that the brain patterns in response to these very different tasks are correlated in the patients. They're more similar, right? They're less unique, right? Whereas the controls are sort of more unique patterns. So this then starts to make us think about, cognitive efficiency, and are we using specialized brain regions, in one group and less specialized in another group, et cetera. This kind of information is completely invisible if you just analyze the tasks separately from themselves, right? And time and time again, this is again work from Jing Sui, we found that by bringing together data, we can get better prediction results, right? And so these are showing some of the regions. Again, temporal lobe and default mode were kind of at the top of that list. But not only that, we can also use that to say, well, this is a lot of data, and maybe we don't want to collect all of it. Let's just find out what's the most predictive, and we can do these ranking things, right? We can say, well, these things are the ones we really want to focus on, and maybe that tells us something about the disorder as well. In my last couple slides, I'm not going to do justice. We do a lot of work led by Jean Liu on brain imaging and genetics and bringing those together. We're looking at genetics and epigenetics and other things. And essentially, we're taking a similar approach where we have maybe fMRI data, and this is whole genome coverage for single nucleotide polymorphisms. It's sort of a genetic polymorphism, and which in some cases can indicate disease, right? And so we're pulling out patterns jointly from the genetic data and from the imaging data, and also looking for algorithms that can sort of search for links between those two, kind of all in one algorithm, and iterate between them. Just to give you one quick example from a neurologic study, this is Alzheimer's disease and mild cognitive impairment. What we did was we found a structural brain, or a volumetric pattern here, that was linked to 4 different genetic patterns. They're linked in different ways, right? And I'm not really going to give you all the details on here, but each genetic pattern has a set of genes and a set of putative functions associated with that gene that can help us unpack information about what's going on. And it also helps us to separate the groups here. So we can see this is looking at 3 different colors, one for controls, MCI, and Alzheimer's, right? Over here we see the structural network, and here are the genetic networks that we found. Okay? And lastly, we don't just have to focus on disease. We can focus on things like cognitive scores, right? And sort of looking at, there's a framework that the NIH is promoting called research domain criteria, which is trying to look at sort of cognitive domains and see whether different combinations of impairment in those domains are resulting in manifestations of different kinds of disease symptoms. And so we can already do quite well in predicting cognitive scores. But that's not the only thing we can do. Once we get this model, we can go back to the data and say, well, let's look at social cognition and see which regions are implicated, associated with that processing speed, visual learning, working memory. And we get to see these very detailed maps of which regions are engaged in these different domains. So in summary, last slide, where do we go from here? I think more work is needed. But just to kind of step you through a few of the areas where we need to do more work, I would say we need to really figure out diagnosis, right? So a lot of people would say we're classifying to the wrong thing. We're using disease categories that are based on symptoms. They're not based necessarily on biology, right? So we need to refine that, looking at those boundary cases. We need to focus on specificity, not just looking at patients versus controls, but looking at other disease groups and trying to really look at sort of the whole story. Is this something specific to that disorder, or is it kind of a general impairment? In terms of translating to the clinic, I think there's a lot of work that needs to be done there, but we have to ask the right questions. What is going to be helpful for a psychiatrist, to know as they're interacting with their patients about this disorder? Some things are not that useful, right? And as we work with that, I think we can develop more of these trial runs, like I mentioned before, where we actually test it in the real world and see if it actually works, see if it can help, and also look at it across multiple sites. So we deal with the scanner effect, for example. Focusing on individual subject predictions, right? Personalized medicine is the buzzword there. And I haven't spent a lot of time on this, but it's a really important item, really trying to get at mechanisms. We ultimately want to know what is going on in this disease. We don't necessarily only want to know it changes these things, right? We kind of want to understand why, And so using the imaging data gives us clues in certain regions that we can connect with certain models that we can then, it's sort of a big circle and go around and test that model in some new data. So that's what I have. We try to practice open science in the lab, and so we try to release our software tools. And as Juan mentioned, there's been quite a people that have used our tools, and we have listservs and do training workshops, et cetera. And last but not least, my lab, whom I'm sure haven't given nearly enough credit. Without them, none of this work would get done. And thank you all for your perseverance and for listening to my talk. So, yeah. Okay, so what do we have? Some question time. Yes. Okay.
To what extent is your work and the effectiveness of your work dependent upon the reliability and repeatability of diagnosis by psychiatrists?
It's very dependent at the moment, right? So that's what I was getting at, refining the diagnostic categories. And so we are trying to get at that in different ways, looking at boundary cases, but also, for example, the diagnosis, you get more confident over time in it. So we can look at these, these imaging data early on, and then if things change, we can see whether that was predictive. But yeah, so that's an issue. It's circular, right? So we need, we need to kind of work back and forth to improve. And as we get better, we're only able to train to what we can target. There are techniques we use called unsupervised learning, where we just let the data try to tell us. We have some cases that we're sure about, and we can train on that in sort of a semi-supervised way. And the folks that we're less sure about, we let the data tell us. But
just a follow-up, if I might.
Of course.
How much resistance are you finding in the psychoanalytic community to these techniques?
I haven't tried to push it too much on that community in particular. And I think we'll have to see. The gold standard is going to be whether it works, right? Whether it helps people. And I think, again, doing these selective trial runs will lay some groundwork. And I think it's going to have to move slowly. It's going to be a change in culture, over time. But ultimately, if it works, I think it will be adopted. So, yeah. Yeah.
Yeah, you mentioned mechanism and translation to psychiatry. I wonder what can be said so far, at least on your work, about this.
So, we know some things about schizophrenia, but right now we actually don't have any really good models. There's been about, I would say 5, 6 different models proposed over the years. And one of the problems is that almost everything is impacted, right, in the brain. So it's really hard to get specific at it. I think one of the ways we're trying to get at that is to highlight sort of what are the most impacted regions, and what do those regions do, and how do they interact with one another, and then say, well, what do we know about dopamine and other kinds of chemicals? We can something I didn't mention here is Juan uses spectroscopy a lot, so he tries to look at the metabolites in the brain, and that's another very important piece of this puzzle. So I don't have a good answer for you on. There's been a lot of proposed models, I would say none of them has really risen, to the top, and I think we're still trying to figure out what a good model is. Yeah.
The test subjects, do you differentiate between the ones who are taking medications and the ones who aren't?
Yeah, medication I didn't mention is really important. So we've done some studies where we've looked at, we can look at basically how much medication they're taking and whether these, whether it's related to that. Some other studies where we have people that are medication naive, right, or they're switching medication, so we can kind of get at it that way. Again, we don't want to just predict you're on a medication, right? That's not useful. So that's a very important question for sure. And there also are a number of studies that are looking at individuals sort of prior to being diagnosed or very early on before the medication has really gotten into the brain. And combining all those things together, we can kind of tease some of that apart. So.
I was interested in the incorporation of genetic data into your very complex data sets that you have here, and wondered if there's much known about the genetic basis of some of these traits you're trying to diagnose, and your expectation for what genetic data may offer with regards to the ability to diagnose.
Yes. So we're still trying to figure all of that out, but there have been a number of studies that have looked at things like heritability, right? So some of the brain networks that we're looking at, default mode network being one, is highly heritable. And there are also a number of family studies, right? So that data set that I mentioned when we're looking at schizoaffective, and we also have unaffected relatives in that study. And so we have genetics, and so one of the things about schizophrenia is genetics is a big factor, but it's not the only factor. So your identical twin, if you have schizophrenia, then they have a 50% chance, of getting about half the time, but it's not 100, right? So there's both environmental and genetic factors, and so that's one of the reasons we're trying to marry these things. Unfortunately, it's not like Huntington's. It's genetically, it's very complex. I mean, these huge studies, they're finding very, very tiny effects spread across, hundreds and hundreds of regions, right? So it's not going to, it's just as complex or more than the brain piece is. So, yeah.
So you're working with large data sets, extremely large data sets, and I was wondering what the criteria is for reducing the dimensionality when you're working in principal component analysis and then converting into...
Right. So it depends on what kind of analysis we do, but we usually use information theory criteria to sort of try to make an estimate of what that dimension is. Or sometimes we go, so I didn't get into the algorithm details here, but we do a lot of try to compress that data down, a lot. Sometimes we go a little bit higher, right? Because we want to basically see, we'd rather not mix things together. We kind of want to split them apart, and then we can remove the noise at a later point. So we often use different criteria for estimating those dimensionality.
I'm sort of interested in the spoilers. So,
The what?
In the spoilers.
Spoilers.
So, for example, back in the day when you applied ICA to bring in data that were cutting edge, and since your connectivity and dynamic connectivity and data fusion, it's like you've always been leading the field. So, what's next?
Well, good question. There's a couple things we're working on. We're doing a lot of work in the deep learning area, really to try to, the problem is that, those take tons, millions of Twitters, right? Things like that to train these complex models. We don't have as much brain data, but we're trying to develop techniques to simulate data to get close, and then train it from there. We're also looking at, in the genetics piece, what we've been doing is combining the static maps with the genetics, and we're looking at really incorporating models that can handle time, space, and subject and genetics all together. So that's another piece. I think we've already got some clues that that's a pretty interesting direction to go in. there's this huge study from the Psychiatric Genomics Consortium, and from that, they had like 30,000 plus data sets and over 100,000 controls, I think. And so they developed these risk scores, genetic risk scores, and we see the risk scores are highly correlated with some of these dynamic connectivity features that we're looking at. So we're looking at models that can kind of do that in a joint framework. And then lots of data fusion is still ongoing, but yeah.
Yes, I was just wondering, back to the imaging with schizophrenic patients, I don't know, do you or are you able to distinguish imaging patterns between patients that have more negative symptoms versus patients that have more positive symptoms? Because I know from a clinical standpoint, the negative symptoms are much more difficult to understand or treat compared to those that have more positive.
Yeah, so that's on the 2 slides I decided to pull out. But yeah, in some cases we can. So one thing we were looking at is for the dynamic connectivity, we can calculate all sorts of interesting measures from that. And so one thing that we calculated was called a hub state. So it's basically they enter that particular pattern of connectivity, and they kind of get stuck, for longer, than they do, other states, right? And so schizophrenia patients have many more hub states than they kind of get stuck in certain states versus controls. And the duration that they spend in that correlates with negative symptoms. And so that's one measure. And there's a lot of different findings that we found, but part of the problem is that measuring symptoms is difficult as well, and it can change. And so you have to do it, this is why working with Juan has been great, because he's done a lot of work in trying to characterize these patients and get reasonable symptoms close in time to when we do the imaging. So, yeah. Great.
Okay.
Oh, we got one more.
I'm just wondering if your data indicates at this point, if you speculated about how to apply this to treatment, for example, how your data is saying that certain symptoms of schizophrenia, for example, could be ameliorated with certain applications, electrostimulation, anything like that. Have you begun that?
I mean, I've thought about it, and it's kind of like that trial run, right? Where we don't want to engage with the patient until we're sure that it's going to work well, right? But so that's why I want to get some more confidence with these trial runs, and if we see things that work, like in the case of working with my Toronto colleague, that actually would have helped someone if it worked, right? If we knew it worked and she'd given us their brain scan, we could have said, hey, you might want to take a look at this diagnosis more than that one, right? There's an ongoing study, speaking of stimulation, we're looking at, one of the projects from one of the investigators is looking at auditory hallucinations and something called, direct current stimulation. You're basically putting like a 9-volt battery on your head and seeing whether that's going to help with those kinds of symptoms. So that's an ongoing study. But yeah, good question. So.
Okay, let's thank Vince for a fascinating lecture.
Thank you.
This work really represents a great body of effort and accomplishments, and it's truly a tour de force by an incredibly prolific researcher. So thanks very much, Vince. My name is, let's give him another one. My name is Gabriel Lopez. I'm the Vice President for Research here at UNM, and my office hosts this event. It's really nice. I've known Vince for a little bit more than a decade now, and it's really nice to see how far this research has come and how far Vince has come, and the impact that he's had both globally in the field of science and technology in medicine, but also locally with respect to the infrastructure that he's helped build in his research area. I think it's fantastic. It is particularly noteworthy and significant for me because when I first met Vince, we were just starting to develop formal biomedical engineering programs here at UNM. And so it's really nice to see that Vince has really carried this field forward for New Mexicans, and so New Mexicans now can fully participate in this area, not only of research but education. At a higher level of specificity, I think it's great that UNM can be leaders in an area that's been identified as a grand challenge for science in humanity, and this was enunciated basically in the form of the BRAIN initiative by the last presidential initiative. So this is great. So to conclude, I'd like to close by presenting Professor Calhoun with a citation in recognition of his extraordinary work and contributions to UNM. And I would also like to personally thank him for his efforts in enhancing our community in research and teaching. And so somewhere down here, I have a plaque.
Oh, wow.
It's, I'm supposed to pose with you.
Okay, sure. Oh, thank you. Yeah, thanks so much. It's great.
Add that to your many accolades.
Yeah.
So thank you all for attending tonight's lecture and joining us in honoring the achievements of Professor Calhoun. I would like to offer special thanks to the members of the UNM Faculty Senate's Research Policy Committee who worked diligently and reviewed many outstanding nominations this year to select Vince as tonight's lecturer. And then finally, I'd like to thank the staff of the Office of Vice President for Research for all their effort in coordinating this great event. So thank you very much for coming. Have a good night, and we'll see you again next year.